Hi, I'm Adrian,
Hi, I'm Megan.
and the enzyme we chose was polyphenol oxidase, and we investigated the inhibition of polyphenol oxidase by sodium chloride.
All right, so the exact study that we were looking at was inhibition of apple polyphenol oxidase activity by sodium chloride, which was conducted by Liu, Lo, and Feng. Apples, especially pre-cut ones, are a popular snack that are growing more and more popular in the food industry. However, a common problem that they face is that the produce turns brown over time when exposed to oxygen, which leads us to the question of why exactly is that bad? Well, the browning of fruit is problematic due to it discouraging customers from purchasing the fruit, which not only leads to a lot of waste, but it also leads to a lot of costs and profits decreasing from the people selling them as well. Some inhibitors, like ascorbic acid and sodium bisulfate, were used in the past to try and decrease the rate of apple browning. However, a common issue is them either leaving an off taste or causing health problems over time. On top of inhibitors, a sanitizer is often applied to produce to maintain food safety. Many of the inhibitors tested in the past for browning control have been found to cancel out the effect of the sanitizers. So, sodium chloride was investigated because it is used in a variety of applications, such as the disinfection of food products and purification of drinking water. Past experiments conducted by the people who wrote this paper revealed that sodium chloride slowed down bacterial growth and the rate of browning of fresh cut apples. If sodium chloride is an inhibitor, it may be a chemical that can act as a dual control agent for produce, browning, and microbial growth. The overall objective then is to find if sodium chloride can inhibit the enzyme polyphenol oxidase to slow down the rate of browning in produce. All right. So, what is polyphenol oxidase and what exactly does it do? Polyphenol oxidase belongs to a group of copper-containing enzymes that is found in many animals, fungi, and plants. This enzyme catalyzes the O-hydroxylation reaction of monophenols to O-diphenols and O-diphenols to quinones. Quinones are reactive when exposed to oxygen. So when produce is cut, damaged, or bruised, over time, the produce turns brown, which leads to the problem that I discussed in the last slide.
So here we have the basic mechanism of polyphenol oxidase. Polyphenol oxidase is found in most plants and animals, and plants specifically, it is found in the chloroplasts or the leucoplasts of storage cells. So here we have an apple. Looking at the apple, it has chloroplasts, and in the chloroplasts, we have a polyphenol oxidase enzyme and colorless monophenols. When the copper complex of polyphenol oxidase catalyzes a reaction between oxygen molecules and the monophenols, this leads to the production of a colorless bisphenol. This will then interact with polyphenol oxidase again and another oxygen molecule to form a colored quinone. When the colored quinone interacts with amino acids and oxygen molecules, this will lead to polymers, which will become polymerized and lead to the production of darker apple melanin.
All right, so next up, I will talk about the structure of polyphenol oxidase, specifically the one found in Malus domestica, which is more commonly known as red delicious apples. All this information on this slide, including the structure on the left, has been obtained through Molstar, which is a bioinformatics website. So polyphenol oxidase has a molecular weight of 56.7 kilodaltons and is composed of one chain that contains a sequence length of 506 amino acids. Its common ligands are copper T plus and oxygen. Now the image on the left right here shows the different colors which represent its secondary structures. As you can see, polyphenol oxidase is primarily composed of alpha helices, which is highlighted in pink, and a couple of beta sheets, which is in yellow. Now, if you look really closely, you can see its ligand binding site, which is shown right here with this green molecule. This is where the mechanisms begin, as Adrian was discussing.
So now on to the materials and methods. The first method that they conducted was the extraction of polyphenol oxidase in red delicious apples. So what they did first was they froze red delicious apples and they extracted the pulp from them. Um, and they mixed this with acetone to form a homogeneous solution. They then vacuum filtrated the solution to form a white powder acetone polyphenol oxidase powder. And the next method that they conducted was an assay method. So in their standard, they had sodium acetate buffer, sodium chlorate, catechol, and TPO extract from the extraction of the polyphenol oxidase. Um, the volumes remained consistent for all cuvettes, and they measured this from 425 nanometers for 3 minutes in a spectrophotometer. The following method that they used was the inhibition of polyphenol oxidase by sodium chlorate and pH. In their standard, they had the same mixture, but they varied sodium chlorate concentration with varying pH and sodium acetate buffer. They found out that the optimal activity for polyphenol oxidase was at a pH of 5 and catechol oxidation at a 50 millimolar concentration. Next was a kinetic study. They had the same standard, but varied catechol concentration and varied sodium chloride concentrations, both which were in 0.2 molar sodium acetate buffer at a pH of 5. They used the same reaction mixture as the standard, and they measured kinetics. Lastly was the pre-incubation of sodium chlorate with polyphenol oxidase. So what they did was they mixed a series of sodium chlorate concentrations with polyphenol oxidase, and they incubated it at 25 degrees Celsius in various times. After this, they added catechol and investigated the kinetic study. And for the scanning studies, they prepared different concentrations of sodium chlorate and sodium acetate. They then added polyphenol oxidase and catechol into the mixture. And then for another study, they replaced the inhibitor with ascorbic acid and sodium bisulfite, and they measured activity from 220 nanometers to 700 nanometers at varying times. And this was done to compare the sodium chloride inhibitor with the new ones.
All right, so next up, we're going to be talking about the results. So we have sodium chlorate and polyphenol oxidase, which was mixed together, as Adrian was saying. And this produced an inhibition kinetic study, which in turn gave us the Lineweaver-Burke plot. The Lineweaver-Burke plot is shown right over here, and it is showing different concentrations of sodium chlorate along with polyphenol oxidase and just its normal ligand. And you can see how sodium chlorate impacts the polyphenol oxidase reaction as it changes the Vmax, which is on the y-axis, or which is the y-axis, the y-intercept in particular, and then the x-axis, which is showing that the Km is being affected. This is suggesting that sodium chlorite is a mixed inhibitor. And then they also wanted to look at sodium chlorite and how the concentration of it and pH conditions impacted the reaction, which generated this graph right over here. Overall, it was consistent that with all the concentrations tested, that as concentration of inhibitor increased, the relative activity decreased. And when it came to pHs, they tested the pHs between 5.5 and 4. 4 resulted in the lowest relative activity, while 5.5 resulted in the greatest relative activity. This suggests that sodium chlorite at higher concentrations and the solution at lower pHs result in the greatest inhibition. Next up is the incubation experiment of polyphenol oxidase and sodium chlorite, which generated the graph right here. They tested different concentrations with the incubation, and it is very consistent that as the incubation period increases, relative activity decreases, suggesting that the longer period of time of incubation, the stronger the inhibitor is. Now, the last experiment that they did was the comparison of ascorbic acid, sodium bisulfate, to sodium chlorite. That produced the graph over here. Now, ascorbic acid and sodium bisulfate, when added to the enzyme solution, automatically rendered itself colorless. As you can see, the absorbance for the two started off very, very low. Now, that is done by them interacting with the quinones. Sodium chlorite, although it took a while, as it was incubated, it decreased in absorbance, which suggests that it may also alter the quinones formed in the reaction. All right, so the main takeaways from the results are that polyphenol oxidase activity is both pH and sodium chlorite dependent concentration, that means. And then sodium chloride is a strong inhibitor that shows mixed inhibitions for polyphenol oxidase. These results suggest that sodium chloride shows potential for acting as a dual control agent for the browning, in order to control for the browning of freshly cut apples and to help with the sanitation of them as well. These findings are beneficial because that would mean less chemicals can be used on the produce, which can decrease the costs and help with the health aspects. Additionally, a dual control agent would lead to a decrease in food waste and a decrease in the profits being lost from the people in the food industry. Future studies can investigate if sodium chlorite can be used on other produce types, and if it is acting as a dual control agent for apples, if it may be able to act as a dual control agent for other produce types, which would be a game changer for the food industry. Thank you for listening. Here are references. If you have any questions, please comment down below.